My name is Dr. Audrey Lafrenaye from Virginia Commonwealth University, and today I'm going to discuss some of our recent findings about the effects of buprenorphine slow-release lab acute administration following a diffuse traumatic brain injury on the diffuse pathology therein. And before I get too far into the talk, I would like to acknowledge the people who've done the work, both within the previous lab members and my current lab members. Everyone highlighted in red has contributed to this work, and Jane Rue was a master's student of mine, and Philip Stone was a medical student. They co-first authored a paper in 2021 that was focusing on our acute studies, and Jane has first authored the paper on our chronic studies that hopefully will be coming out later this year. As many of you know, traumatic brain injury is a serious healthcare problem with millions of people suffering a brain injury globally every year. Hundreds of deaths in the United States are reported every day, and 15% of U.S. high schoolers have reported sustaining a traumatic brain injury within the last year, and this doesn't even incorporate the unreported cases of brain injury. So the pathologies associated with traumatic brain injury come in two general flavors. There's a focal injury, which is usually involving a direct impact of the brain on a solid object, either the craniovault or an outside object. This damage is usually very homogeneous within that pericontusional lesion. There's also diffuse injury, which is more widespread. It can also be localized, but particularly the cellular response is very heterogeneous. And while studies in humans indicate that these diffuse pathologies play a significant role in the morbidity associated with traumatic brain injury, we still don't know that much about diffuse brain injury, and particularly how analgesics such as buprenorphine could impact the progression of these diffuse pathologies. Therefore, my lab specifically focuses on diffuse injury. And to do this, we use adult male Sprague-Dawley rats, or at least in the study I'm going to show you, it's only adult male rats. And we utilize a central fluid percussion injury model presented here that allows us to produce injuries very similar to what we see in the human population, where we're not getting gross pathological changes in the brain, but we do see microscopic changes that are very similar to what we see in human postmortem tissue. And particularly within this study, we assess behavior prior to injury and at 4 weeks following injury. We also assess the physiology of the animals throughout the pre and post-injury immediate time point, and we administer our buprenorphine slow-release lab subcutaneously at 1 milligram per kilogram in our animals at 15 minutes post-injury, and we assessed our pathology at 1 day and 4 weeks following injury. And so the first thing we wanted to know, because buprenorphine has some impact on the heart rate and other systemic physiology, we wanted to look at whether or not physiology was affected by bup-SR lab. And so for a 1-day study, we looked at only traumatized animals, and we saw that the blood oxygen saturation and the heart rate was consistent. We also saw that the intracranial pressure, the pressure within the head, was also consistent between our saline-treated black bars and our buprenorphine-treated red bars. 4 weeks following injury, we also saw consistent blood oxygen saturation and heart rates in our animals, as well as consistent intracranial pressure at 4 weeks post-injury. Now, you might notice that the 1-day study only has 2 bars, whereas the 4-week study has 4 bars. In the 1-day study, we only looked at injured animals with saline or with buprenorphine, whereas in our 4-week study, when we didn't actually anticipate seeing anything, in our 4-week study, we then incorporated sham animals that were treated with saline or buprenorphine. So our solid lines are our injured animals treated with, the black lines will be saline throughout the talk, red lines will be buprenorphine, and the dotted lines are sham animals. Again, black dotted line is saline, red dotted line is buprenorphine throughout the talk. So for the pathology, we wanted to focus on two different brain regions. When injuring using the central fluid percussion injury model, the lateral neocortex is particularly impacted by this particular model of diffuse brain injury. Then also the thalamus is particularly impacted. We were interested in looking at both brain regions because both the cortex and the thalamus are highly involved in human traumatic brain injury. So the first thing we looked at 1 day following injury is diffuse axonal injury, which essentially when an axon is injured, the axon itself will detach, leaving a proximal bulb where proteins like amyloid precursor protein can pile up. And so we're able to visualize that early following trauma as these little brown dots. So when we assessed the amount of axonal injury within both the cortex and the thalamus in buprenorphine and saline treated animals following trauma, we did not see a significant difference between those two groups in either region. We also looked both in our acute study, but what the data I'm showing you is our 4-week study. We looked at the percent or number of total cells to look at whether or not we were getting any cell loss. This model traditionally does not have cell loss, but we weren't sure whether or not buprenorphine would compound that. So we looked at total number of cells within the cortex and the thalamus. We saw that while there was a significant increase in the number of cells in the thalamus compared to the cortex, the region is a little bit bigger, makes sense. There was no significant alteration in association with buprenorphine. We found the same thing, no effect of buprenorphine when looking at the percentage of damaged neurons that had pH changes that we could pick up on hematoxylin and myosin assessments, pycnotic nuclei, and eosinophilic cytoplasm. While we did see some, we did not see a significant alteration with buprenorphine. We next wanted to look at microglial activation, and we do that by looking at the morphology of the microglia. Non-active resting microglia are usually very ramified with a lot of long, highly branched processes. However, as they become more active, the processes become thicker and fewer. So we assessed the morphological details of these microglia at 1 day following injury in cortex and thalamus. And we specifically found that within the cortex, and again, these are all injured animals, there was a significant reduction in the process network of the cortical microglia that we did not see recapitulated in the thalamus, indicating a region-specific change. When we followed these studies out to 4 weeks following injury, again within the cortex and the thalamus of our sham-treated animals, either saline or buprenorphine, and our central fluid percussion injured animals, either saline or buprenorphine, and looked at the morphology of these cells, we also saw changes specific to the cortex. So we see the cortex and the thalamus. The white bars represent sham saline group within the cortex or the thalamus. The gray bars represent the sham buprenorphine treated groups. The yellow bars represent the injured saline treated groups, and the black bars represent the injured buprenorphine treated groups. And we found specifically that the sham injured, so non-injured animals treated with buprenorphine had morphologies that were more similar to the activated morphologies that we see following injury, again within the cortex, but not recapitulated within the thalamus. So we also looked at the astrocyte population, and astrocytes are usually assessed by the size of their cells. They're bigger as they become activated, the amount of area that the GFAP positive astrocytes are covering. And so we assessed that total number of astrocytes per field, the percentage of coverage, the astrocyte cell size, and how circular the cells were to get a little bit of an idea of their morphology, not using as complex assessments as were done in the microglial population. And we found that whereas the microglia were changing specifically within the cortex acutely at 1 day, the thalamic astrocytes were the ones that were primarily changing with larger cell sizes in the buprenorphine treated animals compared to the saline injured animals in the thalamus. When we followed this up in our sham animals and our traumatic brain injured animals at 4 weeks following injury, we found that the thalamus was still incorporated in this buprenorphine associated change in the astrocyte population in terms of the cell size as well as the percent coverage. We finally wanted to look at the third primary glial cell in the brain, the oligodendrocytes. But as these were adult animals, we really wanted to look at myelination because myelin deficits do occur following axonal injury in the case of a traumatic brain injury. So we looked histologically at the myelin fibers as well as the debris. We did not see a significant difference in association with buprenorphine in either time point. We also looked at myelin basic protein, or MBP, and all 4 isoforms at 1 day following traumatic brain injury. We did not see a difference in any of the individual isoforms or overall MBP with buprenorphine. However, we did see a significant decrease in the thalamus compared to the cortex in terms of MBP expression at 1 day. At 4 weeks, we saw a more consistent expression of MBP in both regions, both the cortex and the thalamus, and specifically increases in the thalamus of both sham and injured animals that were treated with buprenorphine. So it appears that buprenorphine has some sort of enhancing of MBP expression, particularly led by the 7.2 kilodalton and the 8.5 kilodalton isoforms. So we also wanted to look at, because diffuse pathology is associated with morbidity, we wanted to do a bit of a morbidity assessment, particularly looking at somatosensory hypersensitivity following trauma. To do this, we do the whisker nuisance task, where we stimulate the whiskers of the animal in an open field, 3 tests for 5 minutes, and we grade a variety of different behaviors. 0 grade is a totally normal behavior. A grade of 2 is a predominantly dysfunctional behavior, with the normal, completely normal score or lowest score being 0 and the highest score being 22. Following sham injury in either buprenorphine or saline treated animals, we found there was not a significant difference between our pre-injury and our post-injury sensitivity. However, as we've published previously, in our both saline-treated and buprenorphine-treated trauma animals, there was a significant increase in somatosensory hypersensitivity. However, that was not altered by buprenorphine treatment. Finally, we looked at weight retention in these animals, and we found that with buprenorphine there was significant increase in the weight retention compared to pre-injury that normalized by 4 weeks, but was also seen at 1 day acutely. Now, I will give the caveat that in these studies, these were all trauma-only animals, and we found that if we, in other studies that we were doing as we adopted Bup as our lab, we found that these animals had a significant drop in their weight at about 1 week, consistent through 1 week, if they were given secondary insults. So in our case, intracranial pressure elevation or an osmotic pump implantation, or both, that we did not see in either our sham or our TBI only studies. So we do not see that if we use a bupe slow release HCl formula instead. And in conclusion, while bupe as our lab did not appear to impact either systemic physiology, neuronal pathology, or somatosensory hypersensitivity, there was a region-specific bupe SR lab associated change in microglia within the cortex and astrocytes within the thalamus. We did also see MBP expression was increased within the thalamus at 4 weeks following either TBI or sham with buprenorphine expression, and that while there was weight retention was better in our bupe SR lab trauma-only animals, which is a study I showed you, that weight loss was an issue in our studies where we had additional insults such as ICP elevation or osmotic pump implantation, that we resolved using a buprenorphine slow-release HCl formula in our rats. And with that, I will thank you for your attention. If you have any questions, I'm happy to answer them, or you can send me an email or find me on Twitter. Thank you.